Hello, my name is Ezonwa Umbunu, and the topic of this discussion is treating cancer with heat through whole body hyperthermia. Now, there are many therapies used in the treatment of cancer, from surgery to chemotherapy to radiation therapy and whole body hyperthermia, and there are also nutritional approaches to the treatment of cancer, such as the ketogenic diet and antioxidant therapy. Now, there's a consensus within the medical establishment that the nutritional approach is not valid, but there's a growing body of evidence, both from the scientific literature and associated clinical trials, that demonstrate the efficacy of this nutritional paradigm. Now, only time will tell whether the ketogenic diet and antioxidant therapy will be integrated into the mainstream of cancer treatment. Now, among the more conventional treatments, certain strengths and weaknesses can be ascribed to each. For example, with surgery, you have the physical removal of the tumor, the excision of the tumor, which is definitely a good thing, because the tumor, which is that community of cancer cells and associated normal body cells, is a highly metabolically active tissue. It uses a lot of nutrients in the body, and it also releases a lot of toxic metabolites that have negative effects throughout the body. Abnormal proteins, certain hormones, certain types of growth factors that just disrupt the fine chemical balance in the body. So the removal of that tumor is definitely a good thing. But the removal of the tumor does not address the underlying cause of its formation in the first place. So there still remains that risk of new cancer cells forming and spreading in the body. Now, with chemotherapy, there exists a vast library of chemotherapeutic agents, ranging from your anthracyclines to alkylating agents to monoclonal antibodies and taxanes. And each of these compounds attack various aspects of cancer cell physiology. But there is a problem because there is a lack of discrimination, in that these chemotherapeutic agents are highly effective in killing cancer cells, but they're equally as effective in killing normal body cells. So in the course of chemotherapy, there is a lot of organ damage in the body, and in many cases, the cancer patient succumbs to the effects of these chemotherapeutic agents and not the cancer in question. Now, with radiation therapy, the area around the tumor is bombarded by a high dose of high energy radiation, and that ionizing radiation damages the cancer cell, it weakens the cancer cell, and even kills the cancer cell. But there's also collateral damage, because neighboring cells receive high dose of radiation, and that high dose of radiation creates genetic instability in those neighboring cells, and it could lead to genetic mutations that might lead to the formation of new cancer cells. So radiation therapy is potentially carcinogenic. And there are also systemic effects, because when the body receives such a high dose of radiation, it has effects all throughout the body, and they're quite negative and quite severe as well. Now, whole body hyperthermia is very interesting in that it damages and weakens cancer cells, but does not really affect normal body cells. So what is whole body hyperthermia? It essentially means raising the core body temperature. Normal body temperature is between 36 degrees Celsius and 37 degrees Celsius. But in whole body hyperthermia, you raise the temperature of the whole body to between 40 degrees Celsius and 42 degrees Celsius. It's just the increment of a few degrees, but it has very, very real effects, especially to cancer cells, because it exploits certain defects in cancer cell physiology. Now, to really understand the mechanism of action of whole body hyperthermia, how this increment of few degrees is able to affect cancer cells so severely, we have to explore a little bit cancer cell physiology. Now, we have a cancer cell, and within the cancer cell is a nucleus, and within that nucleus is genetic material, DNA. Now, cancer cells have mutated DNA because it's the mutated DNA that is its quintessential feature, because its altered nature proceeds from these genetic mutations. And it is these genetic mutations that allow cancer cells to exhibit a high degree of autonomy, because normal body cells are subject to control from hormones, growth factors, and nerve signals. So they are all coordinated, and they don't do anything that they're not told to do in information communicated through these hormones, growth factors, and nerve signals. But cancer cells have a high degree of autonomy. You can say that they are parasites in the body because they utilize nutrients in the body, they release all sorts of harmful waste in the body, and they multiply and spread in an uncoordinated fashion, in a very haphazard and rapid fashion, and they migrate and invade various sites in the body. That independence, that aggressiveness, that virulence proceeds from genetic mutations and the genetic instability that is in the cancer cell. But there is a problem that arises from these genetic mutations for the cancer cell, because DNA is the information of life. It's the instruction for how to construct the machinery of life, which is essentially RNA and proteins. They are the machinery of life. Even though RNA does have some informational properties, and it straddles between DNA and proteins. So there is a flow of information of sorts. DNA is transcribed into RNA, which is translated into proteins. And proteins form the structural components of the cell, and proteins are also the enzymatic machinery. They drive all the numerous, the millions upon millions of chemical reactions that are needed in the living organism. It's largely driven by proteins and some RNA. So the DNA is the information for how to create all those essential products. And when you have abnormal DNA, you also transcribe abnormal RNA, which is translated into our abnormal proteins. So there is a lot of junk proteins within a cancer cell, and it's called a proteotoxic environment because these proteins are not functional. They just form into clumps, and they disrupt the activity of the cell, they occupy space, they disrupt the activity of normal proteins within the cancer cell. So it's just a very disorganized environment within the cancer cell. Very highly disorganized, and even the structural elements show some defects. The cytoskeleton, the skeleton of the cell, also manifests some defects. So clearly, a cancer cell is not this super cell or this very strong cell that is able to subdue the body. In many ways, it's at the very edge of survival, and it's able to survive just because it's able to evade a system called apoptosis, which is essentially cellular suicide. It's able to inhibit that, that mechanism that causes a normal body cell to kill itself. Because a normal body cell, when it incurs damage, it recognizes that it's no longer functional to the body, and it essentially commits suicide. But cancer cells, in the course of the multiple genetic mutations, also acquire a resilience. They're able to shut off the apoptotic machinery, and they're essentially able to carry on with accumulated damage and just keep on trucking along. And in addition to these genetic mutations, they also have dysfunctions in their mitochondria. Now, mitochondria are organelles within cancer cells that are special, that within all cells, that generate energy. And they generate energy in the form of ATP. And they utilize various substrates for generating energy. They use the breakdown products of fat, the breakdown products of sugar, the breakdown products of proteins to generate energy. And they generate energy in the form of ATP. And ATP is essentially the energy currency in the cell, and it drives the protein machinery. So if a protein can be considered a truck, and the fuel of that truck is diesel, the fuel of a protein is ATP. It's what allows it to perform its myriad functions. So there's mitochondrial dysfunction. And this has very real implications for the cancer cell, because cancer cells utilize their mitochondria in an abnormal way, because the mitochondria themselves are abnormal. So they generate more of their energy not in the mitochondria but within the cytoplasmic space. And they use a process called aerobic glycolysis, which is essentially the breakdown of sugar. But the problem with a reliance on the breakdown of sugar for energy is that the breakdown of sugar generates a lot of acidic metabolites, such as lactic acid. So cancer cells also have a highly acidic internal environment. So there's a high degree of acidity. And this high degree of acidity also worsens the problem of abnormal proteins, because proteins are very sensitive to the pH of an environment. And if the pH is not within a certain range, the proteins might tend to lose their shape or might essentially fall apart. The machinery will break down. So there is a real problem within the cancer cell. And the cancer cell is just able to carry on because it's able to subdue that signal that tells a normal body cell to kill itself. So it's able to carry on with all this damage. So where does whole body hyperthermia come into this? Well, cancer cells are able to survive with all this damaged machinery and waste products and all these other challenges that they go through because they upregulate a certain family of proteins called heat shock proteins. Now, heat shock proteins are in all cells in the body, and they serve critical functions in the body, because a protein is a string of amino acids. So a protein at its heart is a linear molecule. It's like a line. It's a string of various amino acids. Now, a protein becomes functional when that string is folded into a very specific three-dimensional shape. It's called a conformation. So when the protein adopts this shape, it now becomes active. Now, heat shock proteins are like chaperones. They help the proteins fold in the right way. When the protein might be needed to be unfolded, it also helps in the unfolding process. In certain times, multiple proteins might clump together. Heat shock proteins help to separate those clumps of proteins. So they are very important chaperones or mechanics of proteins. They help the protein machinery function optimally. Now, in cancer cells, they upregulate their heat shock proteins. The levels of heat shock proteins in cancer cells are much, much, much higher than in normal body cells. And it's much higher because of all the abnormal proteins that they have. The damaged DNA is creating a lot of junk proteins. So heat shock proteins help to normalize some of those junk proteins. And then the acidity, because of their mitochondrial dysfunction and reliance on sugar metabolism, the high acidity also damages some proteins. So they need heat shock proteins to also repair or to make sure that those proteins maintain their shape, even though the environment is highly acidic and it's not, good, and it's not an optimal environment for the proteins to operate in. So they upregulate heat shock proteins. In normal cells, the levels of heat shock proteins are much lower, but the heat shock proteins also perform critical functions in all cells. But what I want you to understand is that cancer cells are at the limit of the function of heat shock proteins. So when you place an additional stress on the cancer cell, there is like a lack of bandwidth. There is no more heat shock proteins to upregulate, that is to synthesize and to release in the intracellular environment, because they're already working full tilt. They can't handle anymore. But a normal body cell has a lot of bandwidth. If it undergoes stress, maybe elevated temperature, possibly UV radiation, or some kind of toxic chemical, is exposed to a toxic chemical, it's able to upregulate its heat shock proteins to help cope with the effects of that stressor on its proteins. But cancer cells lack that ability. And that is what whole body hyperthermia exploits, because raising the core body temperature is a stress on proteins, because proteins are not only sensitive to pH, they're also sensitive to temperature. So when you elevate the body temperature from 37 degrees to 40 degrees Celsius, proteins tend to clump together at that temperature. They tend to lose their shape. They tend to change shape slightly. And a normal body cell is able to upregulate its heat shock proteins and normalize its internal situation. But cancer cells are not able to. So it triggers a cascade of proteins clumping together, more proteins being defective. It overwhelms its heat shock protein system. And that's how whole body hyperthermia places stress on cancer cells. And because normal body cells are able to cope with that stress, especially when you limit the temperature to between 40 and 42, normal body cells are able to upregulate their heat shock proteins and are not affected by that rise in temperature. But cancer cells are placed under a lot of metabolic stress. Now, when this is done repeatedly, possibly 2 times a week, cancer cells have accumulated damage. The next treatment of whole body hyperthermia, there's more damage. The next treatment, there's more damage. The next treatment, there's more damage. So it puts the cancer cell essentially in a dormant state, because the cancer cell is not able to thrive when it's exposed to this elevated body temperatures. And it's not that it's not only able to thrive, it's also placed under a lot of stress. And another critical thing happens when cancer cells are exposed to elevated temperature. Heat shock proteins are typically primarily within the cytoplasmic compartment. They're within the interior of the cell. But when cancer cells are placed under this high level of stress, this additional stress, they redistribute some of their heat shock proteins to their surface. Now this is hugely significant because cancer cells are able to evade the immune system. There are many cells in the immune system that have the capacity to kill cancer cells. For example, natural killer cells, macrophages, neutrophils, cytotoxic T cells, they are all able to kill cancer cells. But cancer cells, through various mechanisms, either subdue the immune system or just make the immune system ignore it. But when you expose cancer cells to this heat treatment, it causes the redistribution of some heat shock proteins to the surface, and this acts as a signal. Immune cells look at this, they're like, this cell is certainly under stress and it should be eliminated. These heat shock proteins potentiate the immune system. Most importantly, the macrophages and the dendritic cells, which are types of immune cells that take up these heat shock proteins, and they now signal to various other immune cells. We have noticed that there is an abnormal cell that is undergoing a high degree of stress, and your body now launches an immune response to the cancer cell that is highly specific to this cancer cell. So how would you perform whole body hyperthermia? Whole body hyperthermia is a very simple therapy. It's performed in some hospitals with highly specialized equipment at very high cost. But what we have to just recognize is the basic principle, which is raising core body temperature. It can be safely performed in a bathtub at home. And what it essentially involves is just, so we have a human being sitting in a bathtub, and the level of the bath water would be at the chest level, with the legs straight in the bath, and the temperature of the bath water would be between 40 degrees Celsius to 42 degrees Celsius, because this is the therapeutic window. This is the temperature range. And a treatment could last anywhere from 30 minutes to 2 hours. But I would recommend starting off with 30 minutes and then gradually, possibly if you perform this treatment twice a week, you start off with 30 minutes, then over time you build it up to 1 hour, and then 1 hour is a good enough time for this treatment to do its work in the body. So you raise the bath water to the chest level, you maintain it at 40 to 42 for that period, so it will require every 10 minutes to check the temperature of the water with a thermometer and then necessarily add hot water and possibly drain some water to keep it within this temperature range. And it's also very useful during the treatment to take in water. And not just only plain water. Plain water is good because it will promote sweating, but you can also, let me say, potentiate this treatment by drinking lime water with black pepper added to it. Now, why this formulation? Because lime water contains a lot of powerful antioxidants. And when you raise the body temperature, the body is placed under additional oxidative stress. So lime water will help to quench the additional oxidative stress in the body and also help the body detoxify some of the harmful metabolites that will be released by the tumors into systemic circulation. So it will minimize any side effect from the therapy. Additionally, black pepper, what it does is that it has what is called a soporific quality. It promotes sweating. And in this whole body hyperthermia, sweating is very, very useful because it will help the body release some waste products, and it will also just help the body cope with the elevated temperature. So black pepper will promote sweating. So it's useful, like let's say every 10 minutes you drink a glass of lime water mixed with like a teaspoon of ground black pepper, and you just drink that every 10 minutes. Make sure you stay well hydrated. It will promote sweating. It will help the body cope with the additional oxidative stress, and it's a very powerful and it's a very useful therapy. Now, I hope you found this talk instructive. Thank you so much for your time.